Glucocorticoids, they are produced from zona fasciculata of adrenal cortex, and little amount of them is also secreted from zona reticularis. Let's know the names of glucocorticoids. Cortisol, it accounts for 95% of the total glucocorticoid activity. It is very potent. Corticosterone, it accounts for 4% of total glucocorticoid activity, and it is much less potent than cortisol. Cortisone, it is equally as potent as cortisol. Prednisone, 4 times more potent than cortisol. Methylprednisone, 5 times more potent than cortisol. And dexamethasone, 30 times more potent than cortisol. The last three, prednisone, methylprednisone and dexamethasone, they are the synthetic steroids. These compounds, glucocorticoids, they have some mineralocorticoid activity too. But the last steroid that I listed here, dexamethasone, it has almost zero mineralocorticoid activity. So if in any therapy specific glucocorticoid activity is required, then this drug is very essential. First, let's make a quick recall of the cellular mechanism of cortisol. The figure shown here is of cell. Cortisol being a steroid, it is lipid soluble. It can easily diffuse through the cell membrane. Once it enters the cytoplasm, it binds with the receptor and forms cortisol receptor complex. This complex enters the nucleus and it binds to the specific DNA region. This specific DNA region is called glucocorticoid response element. Transcription of DNA takes place to form mRNA. mRNA diffuses into the cytoplasm from the nucleus, and in the cytoplasm it is translated into proteins. These proteins are mainly enzymes that take part in the metabolism. Cortisol has the most important role in carbohydrate metabolism. It does two major functions. First, it stimulates gluconeogenesis, and second, it decreases glucose utilization by the cells. The ultimate effect of the cortisol on carbohydrate metabolism is to increase blood glucose level. So how it affects gluconeogenesis? Gluconeogenesis, it takes place in the liver, and meaning of gluconeogenesis is synthesis of glucose from amino acids and fatty acids. From the muscles, amino acids, they are mobilized into the liver. In the liver, these amino acids, they are synthesized into glucose by series of enzymes. This process is called gluconeogenesis. Insulin, it inhibits the enzymes which are involved in synthesis of glucose from the amino acids. So now let's see how this gluconeogenesis process is affected by cortisol. Cortisol, first, it increases the synthesis of enzymes which are involved in conversion of amino acids into glucose. Second function is cortisol increases the mobilization of amino acids from the muscle into the liver. So the ultimate effect is to increase gluconeogenesis. And third effect of cortisol is it antagonizes the insulin's action on gluconeogenesis. Insulin normally inhibits the enzymes of gluconeogenesis, and cortisol, it antagonizes the action of insulin. So again, it stimulates gluconeogenesis. Apart from gluconeogenesis in the liver, next function of cortisol is to inhibit glycogenesis. How? The synthesis of glucose into glycogen is known as glycogenesis. Insulin stimulates enzymes involved in glycogenesis, and cortisol, it antagonizes the action of insulin here. So the ultimate effect of cortisol on the liver is to increase liver glucose. Next function of cortisol is to decrease glucose utilization. So this figure is of cell. Insulin, it acts on the receptor, that is alpha alpha, beta beta, it is a tetrametric receptor. So insulin acts on this receptor. When insulin binds to this receptor, the phosphorylation of insulin receptor substrates takes place, and downstream signaling pathway like PI3K, phosphatidylinositol 3 kinase, they are also phosphorylated, and the actions of insulin on different metabolism takes place through these signaling cascades. So an ultimate action of insulin is also to transport GLUT4 vesicles into the cell membrane from cytoplasm. So cortisol causes insulin resistance. How? First, it inhibits the translocation of GLUT4 vesicles from cytoplasm to the cell membrane. Next, cortisol, it inhibits the expression of insulin receptor substrate 1 and PI3K, which are the downstream signaling pathways of insulin action. So though there is insulin cannot act. This is called insulin resistance. So the function of insulin is to utilize glucose, and glucose will not be utilized. Next is NADH is oxidized into NAD+, and this NAD+ has a key role in glycolysis. Glycolysis cannot proceed forward if there is less amount of NAD+. If there is increased NADH and decreased NAD+, then glycolysis will run in reverse pathway. So cortisol, it inhibits the oxidation of NADH into NAD+. NAD+ will be less and glycolysis cannot proceed forward. So by glycolysis means utilization of glucose. The role of cortisol in protein metabolism, it depends on whether it is extrahepatic cell or hepatic cell. Extrahepatic cell here means muscles and lymphoid tissues. Let's first see what happens in extrahepatic cell. Cortisol, it decreases protein synthesis, but protein catabolism, it continues as usual. How cortisol decreases protein synthesis in extrahepatic cell? First, it diminishes the uptake of amino acids from blood into the extrahepatic cell. Second, cortisol, it depresses the RNA functions. RNA is required for synthesis of proteins. But protein catabolism, it continues. Because of degradation of proteins, amino acids are released in the cell. These amino acids, they are further released into the blood. That's why in blood amino acid level will increase. But in hepatic cell, protein synthesis will be increased. How? First, cortisol, it increases the uptake of amino acids from blood into the hepatic cell. These amino acids, they are converted into proteins by series of enzymes, and these enzymes are stimulated by cortisol. And when liver proteins are increased, these liver proteins will be released into the plasma, and in the plasma they are called plasma proteins. So the effect is increased plasma proteins. So cortisol's function in protein catabolism mainly can be summarized into 4 functions. First, it increases the uptake of amino acids into the hepatic cell. Second, it stimulates the enzymes required for synthesis of proteins from amino acid in the hepatic cell. Third, it diminishes the uptake of amino acids into the extrahepatic cell. Fourth, it depresses RNA function required for protein synthesis in extrahepatic cell. Role of cortisol in fat metabolism. In adipose cell, fat is stored as triglycerides. How triglyceride is formed in adipose cell? First, glucose is transported from blood into the adipose cell. This glucose is converted into alpha glycerophosphate. Alpha glycerophosphate combines with free fatty acids in adipose cell to form triglycerides. But what is the effect of cortisol in this process is, first, cortisol, it increases the insulin resistance, because of which uptake of glucose cannot take place. When glucose is less in the adipose cell, alpha glycerophosphate will be less, because of which it cannot combine with free fatty acids. These free fatty acids, that's why, will be released into the blood from adipose cell. Plasma free fatty acids level will increase. These free fatty acids, they do 2 functions. First, cortisol also increases their oxidation, that is, fatty acid oxidation is increased, because of which energy now is given by free fatty acids instead of glucose. Next is, these free fatty acids, they cause insulin resistance. Now let's summarize the function of cortisol in metabolism. In the liver, liver glucose and liver proteins are increased. In the extrahepatic tissues like muscle and lymphoid tissue, proteins are decreased. In the adipose tissue, triglyceride will be less, decreased. And in the plasma, glucose, free fatty acids and amino acids will be increased, and plasma proteins will also increase.